Um, one minor change to our program this morning is that Dr. Danny Welch is going to be going before me. And that's because in the world of Zoom, he has his own conference that he has to go to. But so to accommodate him and really, we wanted him to, to be at this conference, and see as such a perennial researcher. And I know Danny hates getting, being put on a pedestal, but he is. But we really wanted him to be here this morning. So just one minor little switch we had this morning, but everything else hopefully should be seamless. So with that, I want to get to the crux of everything. I'm going to introduce Danny. Let me stop sharing my screen. Danny, why don't you start putting up your screen before I introduce you? So Dr. Danny Welch, many of you in the metastatic breast cancer community and in the metastasis field knows him. He doesn't need an intro, but I will give him the intro that, for those who don't. Dr. Welch is a founding director of the University of Kansas Medical Center's Department of Cancer Biology. He's also a professor there in the medical school, and he's a director of the NFCR Center for Metastasis Research. His research is in the area of metastasis suppressor genes and the biology of metastasis, and he is best known for his discovery of the 4 of the 24 well-known metastasis suppressor genes. These are basically genes that stop the growth of the metastatic cells, and I'm sure he'll talk about it more. He's a previous Komen Scholar, and he has authored over 140 original research papers in the area of cancer biology and metastasis. And his research is focused again on unraveling the biology of metastasis. And Dr. Welch is an amazing mentor. And I have to tell you, I'm one of those folks that he has mentored along his career, and I'm very thankful for him to be here. And without further ado, Danny, please go ahead.
Well, Josh, thank you. And thank you for being so accommodating with trying to juggle multiple schedules. I am honored to be leading off this morning a group of speakers that you're going to hear a lot of details on. And I'm taking my role as the person who will sort of set the foundation upon which you'll see a bunch of details and exciting research that each is doing. And with that, I'll just sort of help get started. I'm teaching medical students and graduate students, and one of the first things we have to explain is why cancer is so stinking hard to cure. And it's because the cancer cells, the drugs that we're using are not entirely specific to the cancer cells. We're trying to develop therapies that are more specific and don't give all those off-target side effects that are so debilitating. In addition, the cancer cells are not the same. Within a tumor, there are thousands of different subpopulations. Some are resistant, some are sensitive, some are able to spread, some are not. But ultimately, the phenotype or the characteristic of cancer cells that's most responsible for cancer-related deaths is metastasis. And so a couple of little facts, it's that 90% of the deaths or the bad side effects directly caused by the cancer are associated with metastasis. The good news is in 2020, despite it being a weird year, it's not an automatic death sentence, which is unfortunately how it was talked about back as little as 30, 40 years ago. We do not yet have a cure, but there are significant advances, and it's not uncommon now to meet people who are living 10, 20 years post-diagnosis with metastatic disease. Unfortunately, despite the magnitude of the problem, funding has not kept pace with the issue of what metastatic disease is. So the key take home that I'd like to go away with before I start showing the process and the hallmarks is that metastasis and making a cancer are not the same thing. They are related, but they're not the same. And it's not it's not realistic to treat cancer in the same way for the primary tumor or the metastases. And in a way, you can look at it this way. Bob Weinberg and Doug Hanahan defined what they call the hallmarks of cancer. And you can look at that as this big black ball on the side. Doug Hurst and I tried to look at the hallmarks, those characteristics that are present in every metastatic cell. And those are the ability to move or invade, to screw up the microenvironment wherever the tumor cells are, and the ability to adapt and then grow somewhere else. And so those form a subset of cancers, and a subset of the metastatic cells are those hallmarks that determine where the cancer cells will grow and populate. So how do you get these metastases? The cells can spread through any sort of highway system present in the body, through the lymphatics, through the blood, or even across body cavities. In the case of breast cancer, it's the lymphatics and blood or hematogenous metastases that are primarily responsible. And I've generated a series of animations, and I'm going to go through this rather quickly because you have other people who get into the details. But hopefully you'll see part of the reason metastasis has been so difficult to treat and understand. It begins with the growth of the tumor. That's pretty obvious. And the tumor cells are mutate such that a subset of them gain the ability to do what normal cells don't do. In the case of breast cancer, breast cells don't move around the body. But those cells in the breast that have become tumor begin to communicate with other parts of the body, such as the bone marrow. And they do this via these little sacs that contain DNA, RNA, protein that actually mobilize the stem cells that generate the blood cells in our body and many of other tissue types. And those stem cells move around the body, get to other places, and they create what's known as a pre-metastatic niche, kind of like a vacation home for a cancer cell. So they're leaving the urban breast and going to a vacation home in the brain, liver, lung, or other organs. And a lot of studies are taking place to characterize these exosomes and the communication that's taking place. After the cells communicate, they begin to walk or migrate. Just like these dogs and this little baby, they actually have to learn how to crawl. And the difference is that you can crawl by different mechanisms. 20 years ago, we actually used to just think of crawling or migrating as one thing. But over the last couple of decades, the ways in which cells move have been better categorized. Some move as a collective, as you see in the top. They move as a group of cells. I kind of liken this to teenagers at the local mall when we could actually still go to the mall. They move as a pack, disrupting everything in their path. Others move independently. That's in the bottom left corner, mesenchymal. This is a sort of that loner cell, but it's got hyperactive abilities to move. Others are a little bit more subtle. They independently move, but they do so without being noticed so much. They're amoeboid. They're kind of like amoeba. They shapeshift and squeeze through the collagen that makes up the tissue. And still, the fourth mechanism, originally described in the late 1950s for certain white blood cells, but for cancer cells, it's been called entosis. And this is when a mobile cell goes through another cell and appears on the other side. So each of these involves literally tens to hundreds of different molecules that have to be expressed in a coordinated way. Then, as they're doing this, people have been showing that the collagen, here you can see it in fluorescent green, gets reorganized. And you can see it's kind of like a chain link fence here. That's normal. But after the first cells have gone through, the collagen gets realigned and it's put in parallel. And you can see electron micrographs showing this. And what that basically leads to is early cells move very slowly, like pioneers moving from the East Coast to the West Coast. And unfortunately, Josh, this is only at a 2 a.m. LA freeway kind of view, but the first cells that move only a couple of microns, or 1/1,000,000th of a meter a day. But later, after the collagen gets reorganized, they're moving that same distance in an hour. So it's much more sped up. And the cancer cells, again, are manipulating their environment, both by communicating to the bone, but by reorganizing the matrix. Recently, the last couple of years, it's been shown that the tumor cells are recruiting different types of neurons or nerve cells, and those are causing changes in the behavior of the tumor cell. The actual mechanisms responsible for how the neurons are regulating the cancer cells, I don't think are particularly well characterized, but this plays very well into the kinds of studies that Josh is doing and Pat Stieg are doing with regard to brain metastases. Tumor cells also recruit in inflammatory or immune cells. Those immune cells squeeze between the lining of the vessels. They begin to populate the various parts of the tumor, and those immune inflammatory cells can attack the tumor, but also promote the behavior of the tumor. And the ones on the left are macrophages, a type of cell that eats others. And on the right are what's known as neutrophils. These have very weird looking nuclei. And one of the speakers this afternoon, Cyrus Kajar, shared this video with me, but it shows how quickly the neutrophils can attack something that they recognize. In as little as an hour, literally, you have all those neutrophils coming from the blood and attacking. Eventually, those immune cells and the tumor cells form a conga line. And although he's not speaking here, John Condelis and his group have shown that it's an alternating tumor cell, immune cell, tumor cell, immune cell. Think it sounds like a conga line, tumor cell, immune cell. And the immune cells, in fact, are doing a lot of invading for the cancer cell to allow them to move, in this case, into a blood vessel, but can also move into a lymphatic. And as they're doing this, remember the cancers are growing behind them, and they're actually pushing the tumor cells towards the bloodstream. And it's a little like trying to get into a Japanese subway. The cells behind are actually doing some of the pushing to get more and more tumor cells into the bloodstream. And the number that will actually enter the vessels or intravasate can be somewhere between 1 million and 4 million cells for every pencil eraser sized tumor every day. And that's really kind of a depressing statistic. But the good news, if we can take the silver lining, is that we don't see millions of metastases. We really only see very few. It's a very inefficient process. And what's been recognized, and you'll hear more from this from Dr. Kuhn, is the cells, while they're in the bloodstream, can be characterized. One can look at the behaviors of those cancer cells and also the DNA that's in them and released from them that maybe provides information that will help physicians diagnose early, detect early, and treat more appropriately. And here's an example of some of that kind of characterization. It's just one of thousands of different ways people are looking at it. But you can have cells in breast, they're normally epithelial in origin. And there's another type of cell that occurs in an embryo, and they're known as mesenchymal cells. And there's a way during the development of an embryo where these can convert back and forth. And cancer cells can have epithelial, mesenchymal, and then mixed properties. And even as little as 5 years ago, people thought there was an epithelial cell and there's a mesenchymal cell, and they were distinct. Now we know there's at least 100 different variations, 1%, 99, 50-50, and so on. So the complexity just continues to grow. Those cells that are in the bloodstream can begin to make friends. They sort of accumulate a posse. They'll cluster with each other, homotypic, or they can cluster with other things like platelets or white blood cells, or even the material that forms a clot known as fibrin. While they're doing this, they're evading host immunity. They're just escaping the immune cells that are trying to attack them. And they're also changing their properties to hide, like Harry Potter and his invisibility cloak, finding ways in which to just completely hide themselves from the immune system and the shear forces that exist in a blood vessel. Remember, the cells are going, but the liquid that's part of the plasma that goes by them is actually pulling against them, much like the air is pulling on these dogs. And that shear force varies in different parts of the body. And it turns out, for the most part, the cancer cells, when they adhere at a very different places, tend to do so on the venous side because it's a lot less shear. And as they get to a different place, they'll adhere, and then they start to squeeze through the very small spaces between endothelial cells to escape, get out into the tissue, a process known as extravasation. And that's where they encounter that pre-metastatic niche that started a whole lot of this. And some of the pre-metastatic niche are some of those neutrophils that I had mentioned before that eject some of their DNA that somehow the tumor cells take up and recognize, change their behavior, and it allows them to begin to grow. And that can even precede development of new vessels by many weeks or months, so that they basically take over already existing cancer or blood vessels to supply them with nutrients. So that's sort of the process in a nutshell. But I want to just highlight a couple of individual points, because this afternoon you're going to meet the lady who actually started the field of metastasis genetics, and that's Patricia Stieg, or Pat. While she, like I, have been studying mostly metastasis suppressors, there are genes that promote the process. And you'll hear some of that from a couple of the other speakers. But these are things like the accelerator pedal in your car. They're the ones that drive the cells. Suppressors block the ability of cancers to metastasize, and they somehow get turned off in a metastatic lesion. And a new category that's emergingly recognized are known as the metastasis modifiers. These are like the transmission in the clutch. You can have the gas, but in first gear, you can only go so fast. In fifth or eighth gear, you can go faster. And so these are modifiers of both the suppressors and the promoters. And various groups are studying those. We're very interested right now in the modifiers that exist in different races. And this might explain why cancer is more aggressive in certain races of people versus others from a genetic level, notwithstanding all of the socioeconomic issues that exist. And finally, I think all of us, I know everybody who's speaking is 100% committed to the priorities of the metastatic patient. Our goal is to give you a cure and highest possible quality of life. And these were the 5 areas that were identified by surveys and conferences as the top 5 areas that metastatic patients wanted to see addressed. And you're going to hear actually about all of them at some level during the presentations today. We want to make sure that there's complete access to care to give the highest quality and quantity of life. Detect it early, treat it more effectively, and wherever it occurs, and give people the best palliative care possible. And with that, the last thing is, here is what it looks like for cancer cells to spread. One of our goals is to freeze those cells so they can't leave in the first place, or they get halted wherever they are. So Josh, I think I've got a couple of minutes for questions if we have time. Thanks again to everybody for listening, and thank you for the invitation.
Thank you so much, Annie. Yeah, so since you won't be part of our panel, we have a couple of minutes to, if anybody wants to ask Dr. Welch any questions, please put it into your chat box. We'll have at the end, we'll have a moderate, all of us coming together and answering questions at that point. But because Dr. Welch has to leave, we have a couple minutes. We're ahead of schedule. I'm just waiting. Yeah.
So I just want to say, I never ever want to give a talk again without Danny going before me.
Yes.
That was an absolutely beautiful introduction. And it's just so important to really understand how metastatic disease is distinct from the primary disease, and how both the science as well as the therapeutic development as well as the clinic really have to think about this differently, which is really where this whole group is all about. Thanks, Danny.
Yeah, no, and I have to tell you, I think I've learned a lot from Danny, and the analogies that he gives, and while we're waiting for questions to come up, then Peter, that was the whole ultimate, goal of today's, is to make a very educational day, and my talk will be actually showing folks what we actually, in a very 30,000-foot level of how do we do experiments in the lab, bringing folks who have never been in a research lab, bringing them into the lab. And so I think Danny does always a fantastic job, and I learned, much. But let me ask you a question, Danny. Since folks we don't have, so what are your thoughts? You said very in a very 30,000-foot level. What are your thoughts on the different races and the populations that you mentioned, right? For inheritability of cancers and metastasis. You touched upon it a little bit, but I know that's one of the things that questions I've gone previously from the audience.
Well, without getting too deep into it,
right.
We know that there are actually differences. I mean, while we're about 99.99% identical as humans, there are differences that exist between different races. And that's part of the reason we have people from Africa who are darker skinned than me, who has no melanin whatsoever. There are other differences that one can look at. And Kent Hunter, a colleague from the NCI, did one of the more amazing studies, in my opinion, where he actually looked in mice, and he's been mapping genes that distinguish strains of mice, just like we have strains of people, and has mapped metastasis modifiers. We've been looking in a part of the cell called mitochondrion. And those are used by evolutionary biologists to define race and actually find certain differences in a single gene, it looks like at this point, that if you have one mutation, it promotes metastasis, and another, it inhibits it. So I saw a chat pop up, but it disappeared real quick.
It was from Pat Stieg, actually, to all of us. Why don't drug companies find drugs for metastasis?
Oh, Pat, well, Pat knows the answer is well or better than I do, but drug companies have limited budgets, even though we all look at them as being highly profitable. To make a drug costs a couple of $1 billion. And metastasis is hard. And I think even showing that one slide where I talked about different kinds of motility, you have 4 different ways just to crawl. And if the cell can't do it one way, it finds another. And that's part of the issue. The big word is heterogeneity and adaptability. But the other is the clinical trials for metastasis. I don't know that there's still any great consensus on how to design a clinical trial that specifically targets metastasis. That's because most of the drug companies treat the metastases as identical to the tumor cell, just in a different place. And that's not a good assumption.
Oh, great.
And I'm sure
Yeah.
she'll get better answer later today.
Yeah, that's a very great point you bring up. We have one question we'll answer, and then we'll go on to the next just because of time. The question is, can we monitor pre-metastatic niches, and how? Great question.
Yeah, it's a great question, and I don't have a good answer for you, Ting Long. There are people who are doing that in the laboratory. Rosie Kaplan, David Leiden, they're the two leaders, I think. So watch for their names. They are working to characterize the pre-metastatic niche. I think it's possible in a mouse or a rat now, but I don't know that it's entirely possible in people yet. But people are working on it.
Okay, great. Yeah, I agree with you. Okay, great. Thank you so much, Danny. And, really appreciate the wonderful, I think, talk that you gave, because it sets up not only my talk but others coming up on the conference. And it's always great to see you. And,
always good to see you. And I apologize to the others because I really would like to be there for everybody else's talk.
I know.
And thanks for the kind comments as well for all of you.
Thanks, Danny. Okay.
Be safe,
bye.
everybody.
Okay, great. So we're going to go on to now, let me share my screen.